Now let's look at how the nephron processes the filtrate once it passes from the glomerulus into the tubule system. The proximal tubule contains a brush border just like the brush border in the small intestine to increase the surface area for reabsorption. Some of the cotransporter proteins are similar as well, such as the sodium glucose cotransporter and the sodium amino acid cotransporters. The job of the proximal tubule is to reabsorb all of the glucose and amino acids. And remember that this is sodium cotransport. Most of the bicarbonate and sodium also gets reabsorbed here as well. Bicarbonate is converted to H2CO3 and then to CO2 and H2O by carbonic anhydrase, and that can passively diffuse through the membrane of the proximal tubule cells for reabsorption. And sodium is reabsorbed with a sodium hydrogen exchanger. Chloride is reabsorbed by exchanging it for a negatively charged base. Now, these solutes are being reabsorbed isotonically because water is passively being reabsorbed because it's following the solutes. The proximal tubule generates and secretes ammonia, NH3, which acts as a buffer for the secreted hydrogen ions. This hydrogen ion secretion is stimulated by angiotensin II, which causes reabsorption of sodium in the process. Therefore, ACE inhibitors will inhibit the reabsorption of sodium and the secretion of hydrogen ions in the proximal tubule. And what will this do to blood pressure? It's going to lead to a diuresis of sodium and water, therefore lowering the blood pressure. The proximal tubule is also the site where carbonic anhydrase inhibitors inhibit carbonic anhydrase. These are drugs like acetazolamide, and they're inhibiting the reabsorption of bicarbonate, and this leads to the diuresis of bicarbonate and sodium. So by limiting the reabsorption of bicarbonate, we get the excretion of sodium bicarbonate, and water follows. The proximal tubule is also the site of digoxin secretion, and renal insufficiency can lead to signs of digoxin toxicity, such as electrolyte imbalances and hypoglycemia. In summary, this segment, the proximal tubule, accounts 2/3 of all sodium and water reabsorption in the kidney. Now let's look at the next section of the nephron, the loop of Henle, both the thin descending limb and the thick ascending limb. In the thin descending loop of Henle, we have passive reabsorption of H2O, and this section is actually impermeable to sodium. So what is this going to do to the concentration of the filtrate? It's going to increase the concentration of the filtrate. And the water is passively able to move out of the tubule due to the hypertonicity of the renal medulla, which is generated by the thick ascending loop of Henle, which comes next. The thick ascending limb is important because it allows for solute reabsorption while being impermeable to H2O. Now, why is this important? This makes the filtrate less concentrated and helps to generate the hypertonicity of the medulla so that water can be passively reabsorbed in the thin descending loop and the collecting ducts, which we'll come to later. Now, the way that the solutes are reabsorbed is with a very special co-transporter called the sodium potassium 2-chloride co-transporter. And it's named that because it actually reabsorbs 2 chlorides for every 1 sodium and 1 potassium ions. This is a very important co-transporter because loop diuretics, such as furosemide, inhibit this co-transporter, leading to substantial diuresis with loss of sodium, potassium, and chloride, along with water, of course. This segment is also the site for magnesium and calcium reabsorption, but this occurs paracellularly. Magnesium and calcium paracellularly, so it's not through the cells but the gaps between the cells, and about 15% of the sodium is reabsorbed in the loop of Henle. So loop diuretics can have a pretty large effect on diuresis. The next segment is the distal tubule. In this segment, we have a special sodium chloride co-transporter that actively reabsorbs sodium and chloride without passive reabsorption of water. So what is this going to do to the tonicity of the filtrate? It's going to dilute the filtrate. And this channel is stimulated by angiotensin II, leading to sodium reabsorption and increased blood pressure. And it's inhibited by thiazide diuretics, leading to sodium loss and diuresis. And this segment also contributes to calcium reabsorption with a channel that reabsorbs calcium. And this channel can be activated by parathyroid hormone, PTH. So all in all, this segment of the nephron, the distal tubule, contributes to about 10% of the reabsorption of solutes and water. So thiazide diuretics can have a moderate effect on diuresis. The last segment of the nephron is the collecting tubule. This is the last place to make adjustments to the filtrate before it becomes urine to be stored in the bladder. In response to antidiuretic hormone, the collecting tubule cells have aquaporin channels placed into their luminal surface, which allows for water reabsorption. And this leads to concentration of the urine. Also, this segment is the site of action of potassium-sparing diuretics. Potassium-sparing diuretics like triamterene, which act by inhibiting sodium reabsorption. So they inhibit sodium reabsorption and therefore promote sodium loss and diuresis while preventing potassium loss. Now, why will there be less potassium loss? Why would this be less? The presence of the positively charged sodium ions in the filtrate, because the sodium channel is being blocked by the potassium-sparing diuretics, the sodium ions stay in the filtrate. This will lead to the electric potential of the lumen becoming more positive. And because it becomes more positive, this is going to inhibit the flow of the positively charged potassium ions into the filtrate. Now, the collecting tubule is also the site of action for aldosterone. And aldosterone acts by stimulating sodium reabsorption, but it also activates potassium and hydrogen ion excretion. And the sodium reabsorption is going to increase blood pressure, and the potassium and hydrogen ion excretion are going to lead to lower plasma levels of potassium and hydrogen ions. All in all, about 5% of the reabsorption of solutes and water occurs in the collecting tubule, so diuretics that act at the collecting tubule don't have a very strong diuretic effect. Now, this physiology is important, so as you move forward with your studying, look back to this section in your book to refresh your memory about the function of the nephron. It's really important to know for the exam, and it will make you shine in your clinical rotations too. But for now, let's go through some questions to drive things home. Name at least 3 mechanisms by which enalapril, an ACE inhibitor, contributes to lowering blood pressure. The answer is 1. Decreased sodium reabsorption in the proximal tubule, 2. Decreased sodium reabsorption in the distal tubule, and 3. It inhibits aldosterone secretion, which will lead to decreased sodium reabsorption in the collecting tubule. So you see a lot of effects on sodium reabsorption, and this question really shows you how intricate the kidney can be. Enalapril, one of the ACE inhibitors, meaning it prevents the formation of angiotensin II, has a number of important effects. First, angiotensin II normally acts in the proximal tubule to stimulate reabsorption by exchanging it for hydrogen ions. So remember that. There was a sodium hydrogen ion exchanger. Now, less angiotensin II is, of course, going to lead to sodium loss in the urine. And since this happens isoosmotically, it's also going to lead to water loss. Now, the second one is angiotensin II normally acts in the distal tubule to promote sodium reabsorption cotransport with chloride. Remember the sodium chloride cotransporter. So an ACE inhibitor is going to lead to more sodium loss and diuresis here as well. And lastly, angiotensin II normally stimulates aldosterone production, which in turn acts in the collecting tubule to reabsorb water. So aldosterone activates sodium reabsorption. And with less aldosterone, we're going to have more sodium loss. Now, angiotensin II has even more effects on blood pressure, which we'll get to, but you can see that an understanding of the nephron is really important to appreciate anti-hypertensive therapy.